Hello and welcome to my Bio 282 final project. I'm Samuel Nicholsberg, and the original research was titled Two Distinct Phases of Chloroplast Biogenesis During Deetiolation in Arabidopsis thaliana. The news story was titled Researchers Reconstruct the Biochemical Mechanisms of Photosynthesis. The news story provided brief background of the institutional affiliations of the research team. They come from universities of Neuchâtel, Geneva, and Grenoble, to name a few. They also provided intro-level photosynthesis information that consists of what chloroplasts are, where they're located, and what they do, as well as the chemical reaction for photosynthesis. They also provide a brief background on the plant being tested, and it is also labeled as the lab rat of plants, and it, like I said earlier, was called the Arabidopsis thaliana. They provide a overview of the experiment and results. Uh, they stated the etioplast transforming into the chloroplast in less than 24 hours, and that they created the roadmap for how that happens. The news article touches on the experiment and how it would be used in the future, such as herbicide research that does not harm the nearby animals. Next, we have the original work. So the goal of the original work was to determine the sequence of events that occurred during chloroplast differentiation. To do this, they constructed an extremely accurate reconstructions of chloroplasts and the two phases that they're in. They did this by using serial block face scanning microscopy to construct 3D reconstructions of chloroplasts during their differentiation phases. This revealed the chloroplast number, volume, and thylakoid membrane surfaces, to name a few. Then quantitative lipid data was used to complement the structural data, which gave them an in-depth analysis of chloroplast differentiation. That data then gave them the results of the two different phases, and that will be shown in the figure on the x-axis and the y-axis in just a second. This roadmap is a roadmap for a critical process for the growth of the plant, and the research came away with loads of previously unknown material about the two phases. Here's the figure as I previously stated. At the top we have the time of light exposure, and the two phases switch between structure establishment and chloroplast proliferation in between the 24 and 48 hour mark. As you can see, the number of chloroplasts and their increase in the two phases, the photosynthesis related proteins and their change. We also have prokaryotic pathway and eukaryotic pathway relating to the galactolipids. You can also see the cell expansion over that time period. There is also the thylakoid surfacing that you can see over that time period as well, and the ultrastructure of the chloroplast. This was provided in the results section and the discussion section, and is the main point of this article. As for the accuracy of this news story, I would consider the news story to be accurate because of the information they provided, like the photosynthesis and results and experiment information. There is also no misinformation or false information in the news article, so it can't be interpreted as inaccurate. One thing, however, is that each piece of information that I named previously was in the news story was very short and at times very vague. Overall, the news article was a bit short, and for the importance and length of the original work seemed very surface level, and not much could be taken away from it. And finally, we have the work cited. At the top, we have the original article and the link to it, as well as the news story coming right below that, and then we have all the photo creds below that. Thank you for attending my research project final.